Good morning, everybody. I'll get today's lecture started. Today and in the next lecture, we're going to discuss epigenetics. You won't be able to find the content in your textbook because I found that somehow the textbooks have a weak coverage of this part, epigenetics. I feel that is inadequate. Why epigenetics is very important for stem cell biology is because no matter we're discussing cell differentiation or cell reprogramming, all the events are not on genetic level. You know, when cell differentiated from a stem cell toward a terminally differentiated cell, it still share the same DNA sequence. All the cell will have the same, exactly the same DNA sequence. When you reprogram a cell from a fibroblast toward a hard cell, or if you reprogram a cell toward a pluripotent stem cell, nothing changed at DNA sequence level, right? Because it's the same cell, the same nucleus. The chromosomes are still the same. So what happens is beyond the genetics. We have a term called epigenetics to describe that. So in today and in the next lecture, I'm going to cover the most important contents of epigenetics. So to start, I would like to describe a few, observations over the years by the scientific community to support that there is this thing called epigenetics that are very important in determining cell differentiation and all kinds of phenotype that individual will display. On the first slide, I show you this calico cat. You know, some people really like cat. This is really a famous breed. People like to collect this kind of cat. The reason is because they have a beautiful coat color. You know, the patterns are sometimes, some people really like that coat color pattern, but you can't clone the color pattern. You know, previously we discussed the technique called somatic cell nuclear transfer. If you really like a trait, like some cow is really produce a lot of milk, you want to keep that trait, you can clone that cow. Or a dog is really a wonderful dog, you may want to clone that dog. So you can keep pretty much, the most of the traits of that individual by cloning. But here, the coat pattern of a calico cat cannot be cloned. That is because the color code, that color combination or the color pattern in this cat is not because of the DNA sequence. A lot of times it's because of the DNA sequence, like previously when we discussed the chimera. You want the embryonic stem cell to have one color, while the recipient inner cell mass have a different color. So when you have a chimera, you can observe immediately because the coat will have patches, right? That's because of DNA sequencing. But here, in this particular case, the color is not encoded by DNA sequence. Of course, the color, the gene for the color is encoded by some gene, that's DNA sequence. But the pattern itself, the combination of the color, is not determined by DNA sequence. It's determined by epigenetics. Let me explain to you what happened in the calico cat. And to start, this color pattern is only for female cat. It's not for male cat. The male will be either orange or black. The male just don't have this kind of a color combination. The pattern only occurs in female. Why? Because the color gene are encoded on the X chromosome. So here, the capital B indicate a dominant gene that encode a orange color, while the small b indicate a recessive gene that's the other allele encoding a black gene. Therefore, for this heterozygous female, it will have a dominant gene, a recessive gene as the pair of allele. But you know, the female mammals will go through a process called X inactivation, because you know the nature try to keep a balance of the levels of genes. The males will only have one X chromosome, right? It's XY. So the male will only have one X chromosome. Therefore, for female, although they have two X chromosomes during development, one of the chromosomes will be inactivated and become a so-called a bar body. Therefore, most of the genes on that particular chromosome will not get expressed. So it's a gene turn off mechanism, so that male and female each will only have one functional X chromosome. For female, the other X chromosome is inactivated. And because of that, you know, here, as you can see, some of the cells in a female will have inactivation of the paternal X chromosome, while some of the cells will have maternal chromosome inactivation. And that inactivation, X inactivation, is random. That happens once the embryo is implanted. During that stage, you know, during the stage that you isolate embryonic stem cell, at that time, actin inactivation will occur, and that process is random. So we can tell in a female. Basically, the female is a chimera. All the females are chimera. Human beings the same. There are the ladies here, you are chimera because maybe some of your cell in your skin and the other cells in a different part of your body, they have different X inactivation. They use different X chromosome, either from the mom side or from the dad side, but that process is random. It's a random process. That's why, you know, the color pattern cannot be reproduced. It cannot be cloned because it's random. You can never have two cats that have the same color because of the random X inactivation process. As you can see, if the orange gene is inactivated, of course, it's going to create a spot on the skin that's black. And if the black gene is inactivated, and then the orange color will display. Therefore, throughout the body, you're going to have combinations, unique combinations for each cat. So this X inactivation is a kind of epigenetic regulation. So as you can see, the genes won't change. The DNA sequence will remain the same. Throughout this cat's body, all the DNA sequences are the same, no matter which tissue we're talking about. But apparently, they have different color code patterns. The only difference here is because of gene expression is somehow, you know, modulated. So that's the first example. And then second, I want to introduce this interesting observation that tells you epigenetic does play an important role in gene phenotypes. So we have this monozygotic twin. What is a monozygotic twin? Basically, they are developed from the same fertilized egg. So the twins can have two different kinds. One is two eggs get fertilized simultaneously by two different sperm. So that can be the mechanism of twin, or it can be one egg that gets fertilized by one sperm, and eventually, once the cell divided to two cells, each of those two daughter cells eventually give rise to an individual. So that's called a monozygotic twins. So they will have exactly the same DNA sequence to start with, all the material They're from the same egg. Everything is the same. Therefore, in early years, they probably look very similar, right? When they were first born, they look identical. But over the year, one may appear taller, they are shorter, or their skin color, all kinds of things can happen over the years because they may have different experiences of life, and they may prefer different diet, things like that. That all can change, how they look and how they behave, things like that. So if you take samples, if you take the chromosome samples from these two twins and you do an analysis like shown here, something is very interesting. And here on top of this of these two pictures, on top of it, that's the samples from 3-year-old twins, monozygotic twins. You have two colors. It's not the two colors are green and red. So green means unique region for one of the twins. Red means unique region for the other twins. Unique, the modifications. Either it's histone modifications or DNA modifications. There's some kind of modification that's unique for each of these two twins. And then when you try to overlap that two color artificially, virtually, when you try to overlap that two color, at early years, you can see they have a high degree of common places. Like shown here, when the red is overlapped with green, you will observe green, yellow color, right? And you can see most of the regions are indicated by yellow color. That means the modification in early years is quite similar. Therefore, you see that the green color and red color overlap. But later on, when you take samples from 50-year-old twins, still monozygotic twins, and you start to observe now they have their unique modifications, DNA modifications or histone modifications. So one will show red and the other will show green. They don't overlap any longer. So if that two colors don't overlap, that means each individual have their unique DNA modification. They don't share many common DNA modification any longer. So that's the evidence of epigenetics. Over the years, although the DNA sequence are the same, but the modification of the DNA sequence, the modification of the histone that's a part of the nucleosome, a part of the chromosome, now changed. Again, everything happens on top of the DNA sequence. It's not exactly the DNA sequence that are changed. So
I have a question.
Sure, please.
So I just want to make sure that I understood it completely. So for the 3-year-old, so the red and green is blended and like show up as yellow, right?
Yeah. So basically, you use some technique to label the modifications. On one of the individual, you label those modifications as red. In the other individual, you label those modifications with the other color. And now you use a computer to try to overlap, those modifications. And now you see yellow color. That means the modification happens on similar places for the two twins. But later on, you take 50-year-old chromosome samples, and now you do the same thing. You label the modifications in one individual with one color, the other individual with the other color. When you try to overlap it, you see the modification happens in different places of their genome.
Oh, I see. Okay. Thank you. Thanks
Yeah,
so
when you
much.
overlap, you can see individual colors. If the modifications still occur in the same place, you should see yellow color, predominantly yellow color. So you got it?
Yes. Thank you so much.
You're welcome. Please. if you have question, always interrupt me, so we can, we will know what is typical. So on this slide I give you the definition. What is epigenetics, or what is the phenomena of epigenetics? I read. This is epigenetics is heritable alternative states of gene activity that do not result from altered nucleotide sequence. So this definition have three important points here. One, epigenetics is heritable. Heritable? Well, if you're confused why epigenetics is heritable, that is, maybe from the first slide, you go to the first slide. Let's go to the first slide. So this X inactivation happens in early life, but this modification, this inactivation will get passed to the daughter cells, yet the daughter cells of daughter cells, all the cells will share the same X inactivation, right? If we're talking about one ancestor and many daughter cells, those will have exactly the same DNA modification or X inactivation. Therefore, we say it's heritable. So one cell can pass it down to the other cell, of course, it's heritable. And that's the first point. The second point is it's about alternative states of gene activity, or shall we say whether it is about whether a gene is expressed, how high that gene is expressed. Okay, so it's always about alternative states of gene activity. In plain words, it's about whether a gene is expressed or how high or how low those genes are expressed. It's always about the expression of genes. That's the second point. The third important point is epigenetics is not a result from altered nucleotide sequence. So if somebody have a mutation, like the DNA, and A adenine is changed now to a T, to a thymine, that is genetics, because the DNA sequence has changed. Okay, so epigenetics must not involve altered nucleotide sequence. So three major points: heritable, alternative states of gene activity, and three, not related to sequence change, nucleotide sequence change. So I have a third example that is fairly interesting, but I don't know this is as good observation or evidence as the previous two, but since this is interesting, I want to introduce this to you. It is published in Time magazine in 2010, 10 years ago. They found that in Stockholm, the Europe country, Stockholm, there is a small village. That small village have a good record of of their agriculture use every year. They have a good record for many years, hundreds of years. They have like it's a good year. You have a lot of goods collected, harvested, and some years you don't have a good production, agriculture production, things like that. You have a very good record, and also they have a very good record of the people in that village. So scientists revisited their record and they found something very interesting. So they observed that if the grandpa experienced famine, that means they didn't have enough food during a span of their child life, like when they're maybe 7 to 11, they didn't have a lot of food to eat. And then that can result in the grandson to have a long life. That's astonishing. So basically, if the grandpa in his childhood doesn't have enough food, and the grandson will have a longer life. If the grandpa have abundant food, somehow the grandson have a short lifespan because they have many people lived in that village over hundreds of years. So they can test that not only in one generation but in many generations. So that's why this magazine feel it is interesting, and this society Utah Epigenetics also use this as some kind of evidence for heritable epigenetics. They feel there is something in the genome that we haven't identified where in the genome this modification occurred, but that modification certainly is heritable because what happened in the grandpa affected the grandson's ability to live long or short. And interestingly, this only happens in boys, only happens in grandpa and grandson, not in females. So I don't know, maybe there are other reasons that can be used to explain this kind of observation. So that's why I say this may not be the best example. But maybe in the future, they will identify the genes that are responsible and the modifications that are responsible. So we'll decode, longevity as being influenced by epigenetics. So hopefully in the future, somebody will be able to find that out. Now, I give you three interesting observations, and I also give you the definition of epigenetics. From this slide on, let's go to more technical stuff. I want to introduce to you the major aspect or major modifications at epigenetic level. First of all, the epigenetics is about how condensed the chromatin is. Generally speaking, this is the general principle here. So on the left side, I show you a EM microgram. This is a high magnification image of a cell on this right side, and also right bottom corner, you have the nucleus, while the rest is cytoplasm. You see a nuclear pore, and inside the nucleus, there are some regions we call it heterochromatin. Well, there are other regions we call it euchromatin. What is euchromatin? What is heterochromatin? So heterochromatin are the region that lies against the nuclear envelope, and then you see under EM this very dark regions, very dark regions under EM. Well, the euchromatin is the relatively light region. So let's drive deeper to the structure and see what is euchromatin and what is heterochromatin, and I will use this illustration to explain that. So the DNA starts with the double helix, the DNA sequence. People say if you extend the DNA to completely thread-like to be linear, it's going to be surround the Earth. How many times I couldn't remember, but it's long sequence there. But our cell will have to pack it, right? It cannot have that long sequence. It have to be packed to condense the structure so that it can be put into a nucleus. Otherwise, the cell cannot host the genetic material. So the first level of that packaging is the so-called beads on a string structure. So this is the fundamental primary structure of chromatin, that's beads on a string. So the beads is histone. You learned from cell biology, that's histone, and then DNA wrapped around two rounds and a half or something like that, to form the nucleosome, and then between the nucleosomes you have naked DNA strand, right? So that's so-called beads on a string. That's the primary structure, and then it gets further packed to give you the 30 nanometer chromatin. That 30 nanometer chromatin can also be seen in A here. So that you see it is much thicker than what's shown in B. That's the beads on a string structure. So once it's packed, it becomes thicker, of course. So this is how the package looks. And of course, it will get even further packed. Like here, to the extreme will be the mitotic chromosome. In mitotic chromosome, you can even observe the chromosome under a light microscope, right? Because the chromosome now is condensed, it's ready to get separated into the daughter cells. From the fully relaxed DNA double helix to the mitotic chromosome, there's 10,000-fold packaging. So they become much compact at the end when the cell reaches the mitotic chromosome. So you can kind of predict the general principle of epigenetics. If the DNA is totally naked, like shown on the very top, so you don't have any histone associated with DNA, the DNA is all the genes on that DNA will be expressed if you have the correct environment. All the genes will be able to get expressed. And in mitotic chromosome, the other end of the spectrum, no gene will get expressed. So in mitotic chromosome, it's so packed up, no gene will have the ability to get expressed because no transcription factor, RNA polymerase, the required component to transcribe the gene can access genes. The genes is now hidden inside the structure. Okay, so epigenetics is about studying how condensed, how packed that chromatin is. So to make it simple, you can say that. So to study epigenetics, the first tool will be study chromatin accessibility or nucleosome placement. So I would like to use 2 slides to introduce you 2 techniques that are used for studying chromatin accessibility. So we're not studying DNA modification or histone modification. We're just trying to draw a map of where the chromosome is relatively open, where in the chromosome you have totally packed region. Okay, so illustrate that we have a part of the chromosome, a part of the DNA sequence, and you can see some region have this bit on a string, like here, and you have an open region like this strand right here. You don't have any nucleosome, and you don't have any histone here, no nucleosome. Well, in contrast, you have this place. You have a lot of nucleosome packed with each other. So from this map, you can say maybe this naked DNA strand will be easier to be the target of transcription factors, RNA polymerases, other proteins that will bind the protein. Will have an easier time to come to this open region. The same will happen again here. Like if the region is open, the transcription factors and other important components of transcription will be able to come here and bind and regulate transcription, gene expression. And in contrast, if you see this, it's totally packed. Maybe not many proteins can come and do something, regulate gene expression. So that's the very, very rough description of epigenetics, but it is true. So we're going to try to draw a map using some techniques. So I'm going to give you 2 techniques, actually 3 techniques. One is called DNase-1 hypersensitivity. So you have DNase-1, which is an enzyme, right? DNase-1 will be able to digest. Shown here, DNase-1 will prefer to have this relatively naked region, like shown here. So you have a kind of like an opening. It's not as compact as here. You have relatively open space, and here you have wider open space. So DNase one prefer to digest right there when you don't have nucleosome. So DNase one can help you to identify the open region. That's why all I want to say. And there is another technique called FAIRI. So FAIRI is formaldehyde-assisted isolation of regulatory elements. So FAIRI is the name. So basically, uh, you use formaldehyde, formalin. It can be used to cross-link DNA and protein. Therefore, the DNA on this nucleosome will be fixed to the histone. The protein and DNA will get cross-linked together. So that's what you see here. But this region, as you can see here, this light blue region, it's not cross-linked to anything. Therefore, when you put it into solution, you can extract this relatively naked region. Use a phenylchloroform to do DNA extraction. What you will extract out is this relatively naked region because the other nucleic acid is bound to the histone, still bound to the nucleosome. So the protein will be in the organic phase. The DNA will go to the aqueous phase. So this is another way you can identify the open region. And the third method I have on this slide is called macrococcal nucleus cleavage. So macrococcal nucleus works differently from the first two methods I introduced. So this is another enzyme, but this enzyme prefer to cut in well-positioned nucleosomes. They like to see nucleosomes somehow. That's just the enzyme always like to see a structure. The enzyme just works this way. This particular enzyme it likes to cut well-positioned nucleosomes. They prefer to cut around the nucleosome, as you can see. It's always around the nucleosome, around the nucleosome. So basically, this macrococcal nucleus will tell you where nucleosomes are. Okay, so the first two methods will find open region, where macrococcal nucleus will find the where the nucleosome is, the relatively compact region. Now I'm going to show you this kind of illustration or presentation in scientific papers. So this is almost always presented like this. So you have the a strand of the chromosome illustrated here. The gene, where the gene is indicated, right? This HAL S5 is the first gene here, and then another gene RBM42 is the next gene. ETV2 is the third gene here. So this tells you where the axon and intron is. Again, where the axon and intron is. So this is the alignment of these genes to the chromosome. You must have read this in other books, but the other important epigenetic information is also plotted. As you can, please don't omit this attack sequence. Attack sequence is an important technique, but I don't want to cover that. So I just want to use it to see this DNase hypersensitivity. So you see a peak here. That means you have a lot. Remember, when you do analysis, it's always a sample. The sample will have a lot of cells, be it a tissue or it's a cultured cell. It's seldom one cell. But nowadays, although it is possible to analyze one cell, but most times you're going to analyze a sample that's many cells. Therefore, when you see a peak, that means, okay, maybe in many cells out of this sample, you have a DNase hypersensitivity around that region. Okay, that's why you see a peak. Well, in this place between peaks, that means no DNase hypersensitivity in that region. So this indicates to you where what regions are relatively open. Okay, so the ferry sequence will give you exactly the same result because both of them will help you to identify the open region. So from here, we can get some information. So later on, I can explain to you. But when you see this kind of peak, you will predict maybe around this region right here, some proteins will be able to bind, therefore regulated expression of House5, ETV2, or RBM42, right? When you have the protein accessibility around this regulatory region, maybe the adjacent genes will be affected. Their expression will be affected. Okay, so this gives you some information about the regulation of those genes. So I'll move on to the second important part of epigenetics. So earlier I discussed how to describe chromosome accessibility. I told you 3 methods. And now I want to go into DNA methylation. Of course, this will have more details, right? Because DNA methylation is at sequence level, while the other sequence which we have just described is about regions, is raw information. Here, DNA methylation will have to take care of the sequence of DNA as well. So yeah, in the field of epigenetics, or maybe to other biologists as well, some people will say the DNA is not made up of 4 nucleotides. It's actually 5 nucleotides. It's not only adenine, cytosine, guanine, and thymine. You have the fifth one. The fifth one is called 5-methyl cytosine. So this is because on this ring here, you have a methyl on the fifth carbon. You have a methyl group used to modify the fifth carbon. Therefore, this is called a 5-methyl cytosine. It's a modification of the cytosine. This happens very, very frequently in the cell. So any cell, you take a sample, you will be able to find adenine, cytosine, guanine, thymine, but you also will find 5-methyl cytosine. So that's why some people will say there are 5 nucleotides, not 4 nucleotides. Okay, so just to tell you, this is very common modification of DNA sequence. Okay, and I just use that to point to you where the modification is. So just some more about DNA methylation. The DNA methylation always happen in the context of CpG. The P is just to tell you that the C and G is linked by a phosphodiester. So this is not it's a sequence. It's not a CP. It's not a CG base pairing. It's not base pairing. So they belong to the same strand. These two CGs is the same DNA strand. It's not base pairing. So that's why they put a P there just to indicate that this is a sequence. It's always in this kind of context. CG, well, the C will be modified by methylation. It's not G, it's C. Cytosine get modified, but always in this kind of context. And generally speaking, methylated DNA is associated with heterochromatin or gene suppression. So as shown here, the methylation is always in this context. It's this cytosine that gets methylated, and then the methylated DNA, generally speaking, is associated with packed DNA. Well, unmethylated is related to relaxed DNA sequence, relaxed chromatin. That's very general summary of the principle. If you were talking about individual cases, it may not be true. Generally speaking, the methylation is associated with heterochromatin or gene suppression, packed chromatin. In the next slide, I'm going to tell you throughout the genome, we have a bimodal landscape about DNA methylation. But here, I just want to introduce two more important concepts here. One is CPG island. The CPG island basically is a region of the chromatin or the chromosome. We call it CPG island as long as that region fulfill this criteria as shown here. So it must be the region must be longer than 500 base pair. Easy to understand, right? It cannot be shorter than that. And the GC content of that region must be higher than 55%. And then in that region, you have a lot of CG. You have a lot of CG in that region. We call it that region as CPG island. So this CPG island usually happen in the promoters. The promoter is the important region that regulates gene expression, right? The CPG island is usually there in the promoters. But here, the second important concept I want to tell you is how to detect the DNA methylation. How you detect that? Actually, the method is very, very simple because you see here we have a DNA strand, and in this DNA strand we have one CG, a second CG, but one of the CG is methylated. So the cytosine is methylated, while the second CG there, the cytosine is not methylated. And now we need to identify the methylation pattern to tell which cytosine is methylated, which cytosine is not. So we just treat the DNA preparation with sodium bisulfate. Sodium bisulfate is a chemical that will convert the unmethylated cytosine to become a uracil. So the cytosine will become uracil after this treatment if the cytosine is not methylated. Well, the methylated one, won't change, will still be a cytosine. Therefore, later on, you just do a sequence or do a PCR, you should be able to detect that because this U in a PCR will be changed to a T, right? Because U happens in RNA, well, the T happens in DNA. So when you do PCR, the PCR is always about DNA. So you change that U to T, while the C remains to be C. So this way, you should be able to tell which one is methylated, which one is not. It's very, very simple, just a chemical treatment followed by either sequencing or PCR. You will be able to tell that information, whether the DNA is methylated or not. So here I want to give you the so-called bimodal pattern. So basically, we have about 28 million CPGs in human genome, about 28 million CPGs, and about the majority of those CPG are actually methylated. They are methylated all the time. And less than 10% of the CPG occurs in the CG dense regions, so-called CPG island, which I have already described in the previous slide. So about 10% of the CPG is in CPG island. But in the CPG island, they are largely resistant to DNA methylation. So the first paragraph says, the majority of the CPG are methylated. But if the CPG is in the CPG island, they generally speaking they are not methylated. So that's the first information. And then here comes the bimodal landscape. In this landscape, it describes that the most bulk genome methylation patterns are static across tissues and throughout life. So throughout life and also across different tissues, the majority of the DNA methylation will not change. It's quite static. It's always methylated. However, in the CPG island, and also in the germline and during pre-implantation development, we see rapid demethylation of the paternal genome at fertilization. And then that is followed by depletion in both paternal genomes during early embryonic progression. So we're going to have that uh, in a couple of slides later. But the bimodal just to tell you the majority is static, they won't change. But in the CPG island, you see quick change, and those change and also have some kind of a temporal regulation. The temporal regulation during early development, you see a lot. You see that the DNA methylation get erased, almost completely erased. So here I give you the several important enzymes that are important for DNA methylation and demethylation. So the enzymes are called DNMTs, indicating DNA methyltransferase. DNA methyltransferase. Some of the methyltransferase performs de novo methylation. De novo methylation means the DNA is not methylated. Now the enzyme can methylate it. It's called de novo, from not methylated to methylated. So de novo methylation. While some other methyltransferase perform the function of maintenance of methylation. That means the DNA is already methylated. The enzyme just keep that methylation status, so it won't get demethylated. So there are two kinds of enzymes for DNA methylation. One is de novo methylation, the other is maintenance of methylation. Well, the demethylation enzymes are quite complex. It's not as simple as the methyltransferase because demethylation takes multiple steps. Methylation is a one-step thing, but demethylation takes many steps. Even here, we don't have all the information of demethylation. So I just give you this description of methylation first. It takes this SAM. SAM is a molecule. I don't know if you're learning about chem or not, but cytosine would take SAM as the donor of the SAM is a one carbon donor, basically. Will give you the methyl group, and the DNA methyltransferase will add that methyl group to the cytosine. Therefore, we have a 5-methyl cytosine, 5-MC, indicate 5-methyl cytosine. But demethylation is first we'll need to have the cytosine oxidized. So the oxidation takes the enzyme TET. A few TET will make the methylated cytosine oxidized. So the oxidation takes alpha-ketoglutarate as the helper here. So you need oxygen, but you also need alpha-ketoglutarate. So now you know the DNA demethylation is coupled to metabolism. Alpha-ketoglutarate is a component, intermediate of the TCA cycle, an important one that give rise to succinyl-CoA, right? That's I believe the fourth step of the TCA cycle. So it takes multiple steps. Basically, first you have this first oxidation, second oxidation, further oxidation. Eventually, it becomes a carboxyl from a methyl group to become a carboxyl eventually, and then this carboxyl will be removed. This carboxyl modified cytosine will eventually get removed by the DNA repair mechanism. So here we'll only show that demethylation will take oxidation, but eventually it will become this carboxyl cytosine. This carboxyl cytosine will get removed by DNA repair mechanisms. So the demethylation takes many steps. So what's important here is when you see a 5-MC, that means DNA is methylated. When you see a 5-HMC or 5-FC or 5-CAC, that means the DNA is this cytosine is in the process of demethylation. So 5-MC indicate methylation, and the rest of 3 means demethylation. So here is what's so-called the 2 ways of DNA methylation during early development. So I wouldn't say two waves. So I show this earlier in our discussion of the somatic cell nuclear transfer for three people, 4 people therapy of mitochondrial disease. I show that again here. So basically, the paternal is more methylated to begin with. Basically the sperm DNA is more methylated, while the maternal, the oocyte DNA is less methylated to begin with. But once fertilization happens, the paternal uh, will quickly get demethylated. While the maternal will also get demethylated, but it happens slower than the paternal side. At the ES cell stage, that's when the cell is in the stage of inner cell mass, it has the lowest DNA methylation. It's not completely erased. There are still some DNA methylation still kept, but it's the lowest in anybody's life. That's when you have the lowest DNA methylation level. After that, the cells will start to differentiate to give rise to all kinds of cells, and DNA methylation will start to pick up. Okay, so here on top is about DNA methylation. That's indicated by the level of 5-MC. Well, the bottom here shows the DNA demethylation. That's indicated by the level of 5-HMC, 5-FC, and 5-CAC. Those are the intermediate during the demethylation process. Okay, so finally, I just want to tell you.
Professor,
Sure. Yes, go
sorry
ahead.
interrupt you, but I cannot see your screen. I'm
Oh,
not sure it's just me or
I can
everybody.
see the screen. I can
I'm
see.
sorry.
No,
I,
I can see the
I,
screen.
my mouse didn't point to the right place. I pointed to the other screen because I used two screen. I'm sorry about that. So, but if I explain this again, in the sperm, the methylation level is higher. In oocyte, the methylation level is lower to start with. But after fertilization, the the sperm DNA will get quickly demethylated, while the oocyte DNA is slower in demethylation, but in the inner cell mass, you find the lowest DNA methylation level. And eventually, the ES cells will start to differentiate, giving rise to trophocectoderm and the 3-germ layers. And now the DNA methylation will start to pick up. So eventually, we will have the normal DNA methylation level throughout our body. And then another important content here is the 5-MC indicate DNA methylation, while 5-HMC, 5-FC, and 5-CAC indicate the demethylation level. Okay, so finally, I just want to point to you how you read DNA methylation in scientific papers. So you have these circles, you get confused. What does it mean? So this paper is from the paper that our student-led discussion will cover. It's from Shinya Yamanaka, the famous study that led to his Nobel Prize and also his, the breakthrough in cell reprogramming. So basically, what he did here, he put 4 factors into fibroblast, and he used this nanog locus as his reporter. So he put this GFP into the genomic locus of nanog. Nanog is a pluripotent gene. Therefore, if you have the GFP into the locus, the fibroblast won't have the expression of nanog. But once the cell gets reprogrammed towards a pluripotent stem cell, the nanog locus will be activated. The cells will show green fluorescence. And now you can pick the green cells and study whether the green cells are indeed reprogrammed from fibroblast toward pluripotency, right? So in A here, he used the markers. Those are all pluripotent markers to indicate the green cells are indeed reprogrammed because the cells express the pluripotent genes, while the fibroblast, MEF, as shown here, largely don't express any of those pluripotent genes. But once the fibroblast is reprogrammed, they start to express those pluripotent genes. But the important thing I want to point to you is this part. You see these tiny circles. What does that mean? So basically, each of this circle will indicate a CPG island. Okay, remember, CPG island is a. I'm sorry, it's not a CPG island. Each block here is a CPG island. In CPG island, you have many CPGs, right? You have many CPGs. So each of this circle will indicate one CPG. And of course, the one the different circle will indicate different CPG within that promoter. I couldn't count here how many do you have, but you can appreciate there are many CPG in the CPG island. Each block is CPG island. So what open circle here means the DNA, the CPG is unmethylated. A closed circle or filled circle means the DNA is methylated. The cytosine is methylated. Basically, that's what it means. So you can see from the beginning, the MEF, which is the fibroblast, the cell you start with, you have the methylation, a lot of methylation in the promoter of these pluripotent genes. OCT34, NANOC, FBX15, they are all pluripotent genes. And in fibroblast, the DNA is methylated. That means in fibroblast, the nature tells the cell don't express the pluripotent gene. They condense the chromatin there. That's why you see a lot of DNA methylation. But once the cell are reprogrammed toward induced pluripotent stem cell, so you do the analysis again, you see that you don't see the same methylation pattern again. All the DNA methylation is removed. Now the cell allow the transcription from those locuses. And again, here, additional information here, as you can see, there are some other genes, like here is let me see, okay. So the ESCs, you see, the H19 and IGF2 receptor are genes specific for somatic cells, for example, like a fibroblast. Therefore, you can see that I got it wrong. So it's heavily methylated. I'm not wrong. Okay, in this somatic cell express the gene in embryonic stem cells is heavily methylated because embryonic stem cell will not express this H19 and IGF2 receptor. Okay, but in MEF cells, you do have some relatively more CPG that are unmethylated when you compare to ES. But look at the iPSCs. I would say they are more similar to MEF than embryonic stem cell. You compare this methylation pattern, you will see they are more similar to the MEFs than embryonic stem cell. So what I'm trying to tell you is that reprogramming is not perfect. If you only study the pluripotent stem cell gene, you will see, okay, it gets reprogrammed. Now the region encoding those pluripotent genes are open. By the indication of DNA methylation, they're open. But some of those other genes, they still have the similar pattern of somatic cell, not the pattern that you want in embryonic stem cell. Because of the task here, you're trying to push the cell toward embryonic stem cell-like. But as the outcome, it's not complete. Okay, that's the end of today's lecture. If you have a question, you can stay behind and ask me. We're going to continue to discuss epigenetics in the next lecture, which is going to be Monday.
Dr. Liu, I had a question about the heterochromatin, because in, like, I think the second slide you showed today, or the first, you showed how it was more condensed near the nuclear envelope. So I was wondering, like, basically our heterochromatin is basically in that region most likely compared to being in the center.
Yeah, let's go there.
Yeah.
Yes. But in the nuclear pore, you see there's a lot of region that's lighter as well. So, it's true. So maybe the cell try to push the genes that not really expressed to the surrounding region. Well, in the center you have more genes that are active. I think generally speaking, that's true.
Yeah, because I was thinking like possibly the chromosome might be interacting with the nuclear envelope, and that also helps in deactivating those genes from being expressed.
I think that's true.
Yeah.
Yeah, that because the proteins in the nuclear envelope will, the packing, as you can see from here, the packing will make the DNA is never 1D,
Yeah, of course.
not 2D, or it is absolutely 3D. So
Yeah.
sequence-wise, maybe in this region, those nucleotides are very very far from each other.
But they can really.
But once you wrap them around, those seemingly far apart genes will now get recruited to the same to the same region and get repressed by the
Yeah.
same mechanism.
Gotcha.
Yeah.
Also had another question about the DNase 1 and
DNase one, yeah.
in FAIR.
Yeah, that's the
Yeah.
Mhm.
So for FAIR, I was just trying to understand. So the whole idea is to extract the DNA and leave behind the nucleosomes, while DNase 1 is to find out the euchromatin areas, right?
Yeah, I didn't tell you the technical details. So usually for this kind of sequencing, you make the DNA to become fragments.
Yeah.
So you have to make them to become fragments. Otherwise, what you get is a single chromosome, right?
Yeah.
Each chromosome will be just the one.
Yeah.
You lose the purpose. You have to make the chromosome to be shorter pieces, and
Mhm.
then this DNase hypersensitivity will when you cut the DNA, you will be able to find where is the breakage point.
Mhm.
By doing sequence, you know where is the breakage point, so you know which region is more sensitive, which region is less sensitive. So I don't understand your question exactly.
Yeah, so I was just okay. So I was trying to understand basically like in FAIR. So what I understand is that it basically cuts off the DNA between nucleosomes, and like in the picture you see there's two cups. One cup has the nucleosomes and the DNA, and then the no chloroform separates the DNA from the nucleosomes. Is that the purpose of FAIR?
Yes, because for FAIR, what you do first is formalin treatment, formaldehyde treatment, right?
Yeah,
So the formalin treatment will make the protein and DNA to get associated permanently with each other.
I see. Okay.
Well, some other the naked region will not have that cross-linking.
Yeah.
So when you do a phenochloroform extraction, you can extract those naked region.
I see.
Yeah,
Okay.
but the cross-linked part, the protein is always in the organic phase, right?
Yeah.
So
Yeah.
to carry the associated DNA in the organic phase.
Oh, okay.
So that way you can sequence the aqueous phase, and now you can tell which region is open. While the
that's
rest, of course, is more nucleosome rich.
okay. That's really cool. That's a nice technique, actually.
Yeah, there are a lot of cool techniques. You can
Yeah.
study ATAC by yourself. So,
Yeah.
so this ATAC, vary DNase1 hypersensitivity, and macrococoa nuclease are raw methods that help you to describe throughout the genome of the openness and the closeness of chromatin.
Okay, I'll make sure to check that out then.
Yeah,
Awesome. Thank you.
you're welcome.
Professor,
Yes,
I had a question about the last slide again. I was just confused by the figure. Um, so
last one.
yeah, is it just
This one.
yeah, this one. So is it just trying to show us that the MEF methylation patterns are just different from other embryonic stem cells?
So you see here, let me explain to you this again. So the MEF is fibroblast, right? It's
I see.
mouse embryonic fibroblast. And then what you're trying to do is you're trying to put transcription factors into fibroblast, and then eventually use this modified genome. You can see that maybe some green cell will appear, and those green cells may be pluripotent stem cells. This is so-called IPS, right? Because you're trying to reprogram the fibroblast toward IPS, induced pluripotent stem cell. So you have two examples of the induced pluripotent stem cell on the first row and the second row. And in the third row is a positive control. You take embryonic stem cell as a positive control because we know embryonic stem cell is the gold standard of pluripotency. Right? So you have three cell types. One is fibroblast, the other is the gold standard, and the third is a cell that you have created.
I see. Okay.
Right? So that's first to understand that, and then let's study these genes. OCT4, NANOC, and FBX15. They are pluripotent stem cell genes. So that means those genes are only expressed in pluripotent stem cells. So that's why when you observe this, their DNA methylation, you can see in MEFs, their promoter CPGs are mainly methylated. You see a lot of filled circles, right?
I see. Okay.
That's because those genes are usually not expressed in fibroblasts. Those are pluripotent genes. They are not expressed in fibroblasts. But once you reprogram the cells toward pluripotency, now you see those methylation are removed.
Oh, I see. Okay.
So they are very similar to the positive control because the positive control is ES cell. The ES cell, of course, will express the pluripotent stem cell gene. Therefore, the DNA are not methylated.
Okay, got it.
So that's the explanation of that part. But lastly, I also wanted you to appreciate something is you can see for these other two genes, H19, for example. Let's don't take H19 as an example. That's not the best example. Let's take IGF-1 receptor as an example. You can see this is a gene not specifically related to pluripotency. It is expressed in fibroblast, but there are a lot of openness in fibroblast. You see a lot of open circle. In embryonic stem cell, you have less open circle, right? When you compare embryonic stem cell and the mouse fibroblast, you see that in embryonic stem cell, you have a lot of filled circle, while in fibroblast, you have less filled circle.
Mm-hmm.
But now let's observe the reprogrammed cell. You see that this also have a lot of open circle. This cell is already reprogrammed. It should be similar to embryonic stem cell. However, I would say if you compare this and embryonic stem cell, you see they're quite different. But if you compare this to fibroblast, I would say, okay, in terms of the number of openness, maybe they're quite like fibroblast.
I see.
What I'm trying to say there is the reprogramming is not perfect. So although you make the cells to gain pluripotency, they may not be exactly embryonic stem cells. They still share some property with the cell origin, which is fibroblast.
Oh, okay. Thank you.
Yeah, that's just one point I want to add there. Not really related to what we're discussing in today's lecture.
Got it. Thank you so much, Professor.
You're welcome.
Professor,
Yes.